All right, I think that's everyone that's going to show up. We're a small community here at ABRF of structural scientists, but we're hoping that the community continues to grow because I think it is very valuable to be a part of. But I am going to start off our roundtable on training and research in structural biology course. And I just wanted to introduce the speakers that we have today. The first is a new colleague of mine, Soma, or Thayumanasamy, Somasundaram. Thank you.
For telling my name.
And he is the director of the X-ray facilities at the Florida State University, and he has over 30 years of experience in training scientists to collect and interpret structural data, and is heavily involved in the ABRF organization as the president-elect of the organization. So you'll be seeing a lot of him in the future if you continue to participate in ABRF. The other speaker that we have today, Joshua Strauss, is a former colleague of mine from UNC, and he's the Cryo-EM Core Director, and he was instrumental in getting the course started and continuing to maintain the core throughout the years. Josh has over 10 years of experience in microscopy and image analysis. Josh is responsible for developing training for Cryo-EM users at UNC and beyond, and notably also has launched a YouTube channel that includes Cryo-EM training resources, which is really cool, and you should check that out. And I am Gabby Budziszewski. I'm going to be speaking as well, just a little bit about my position at the National Crystallization Center as the operations manager. And I want this to kind of be an informal session, so if you have questions during the talks, the speakers have agreed that it's totally fine if you want to ask questions. I want this to kind of be a back and forth so we can build the community. So without further ado, Soma?
Thank you, Gabby. I know we are a small group of people here, and I have twofold interests. I've been a structural biologist for many years, and then also wanted to bring the diversity into ABRF. I mean, if you've seen one of the slides, we are part of the 3% of, maybe a 0.1% of 3% or something, right? Therefore, I am, as Gabby said, I'm the incoming president of ABRF, and I wanted to not only bring the structural biology and also non-biology course into, because we all face the same problems, whether you are an electron microscopist or NMR person or whatever course you run. Some of the problems are common to us, right? Dealing with here, Susan here, she has been running this one in Moffitt for 30 years. She will tell you the problems you see in all the courses are very similar, even though the technology or the discipline may be slightly different, right? Therefore, let me make sure I do this advance. I'm at Florida State. I've been there since 1993. At the time, we started the X-ray crystallography core. At the time, it was the only X-ray single crystal macromolecular crystallography facility in the Southeast, right? People from Florida, Georgia, and Alabama came to our facility. Fast forward to 31 years, we are in a different position, right? I mean, there's also a talk tomorrow. Marcus and I'll talk tomorrow also slightly in this area how the challenges we are facing in structural biology course. Okay, let's get on to today's one. As you all know, I don't have to preach to the choir here. It's a niche core, right? We are running a core which is probably one per institution, right? Because there are not that many cores similar to yours in the institution where you're working. And then we are truly interdisciplinary people, right? Because I'm not saying that other biology courses do not have a multiple disciplinary people. In a practical terms, in my facility or in my institute, we have students from biology, chemistry, mathematics, computer science, physics and engineering, right? These are the people who typically come to me. Therefore, we are truly interdisciplinary science. That has got implication in training, right? Once again, as Gabby said, just stop me, we can, and then we have a unique user base, and then, as many of you know, the project timelines can be very long, because people come to me for crystallization, and it fails, and they do not come to me for some time, and then try again, and then they do the crystallization, and then the project line time. We will go through this. This is some of the areas we want to talk about, and many of you know, many of the biological, structural biology course depend heavily on open source software, right? That has its own challenge because it's developed by, it's very similar to Linux system you are all probably using, and most of them are open source. It has got its own problem. Therefore, as a core facility staff and scientists, we play a greater role, and that is what we are going to talk about, the education and training part of the talk. Right? And as I said, I'm just going to repeat myself here. I deal with maybe 4 or 5 PIs who are using the facility. Therefore, I work with them, and then that is most likely across the many structural biology course is true, right? And then, however, we are needed because we solve the 3-dimensional structures of biological molecule, which play a great role in function and mechanism of biology. That's why we play a greater role in even though we are small in footprint or usage, we play a greater role in biology, because that's what we are interested in, right? Unfortunately, in my case, there's a threshold to get to me that the crystallization has to work. Unlike the microscopists in the audience, you are coming and taking away my thunder because you can use any sample you want and look at it, right? The problem is I have a threshold if it doesn't crystallize. True, but you have to have a crystal. Therefore, there's a modest threshold to get to me. Unlike other cores, this has got an implication as you run the core, right? People need to be successful before they become your customers, right? And then, therefore, it is a collaboration, and cost recovery is hard. And I come from a state university where I'm an employee, therefore I'm not recovering my salary. And because we are a state university, we have certain restriction on what we can charge people, right? It just plays into both the role, right? Of course, there are not that many vendors who peddle the instruments. As you know, this is a multidisciplinary area. We have people coming from biology. That is what drives us. That's what we are wanting to get to. And then, of course, we need people who have biochemistry background, with the people who purify your proteins and make sure they are pure. And also, you need to have some level of physics background, interaction between the electromagnetic radiation biological samples. Therefore, you need to have some level of understanding of how the whole thing works. And then finally, if not more important, is the software. As I talked before, we have a lot of open source software, and then you need some kind of hand-holding to tell how it works, and when it fails, how you help the people. That is where our core facility staff, we play a bigger role. Right? And once again, this is repeating myself here. Molecular biology people are needed to they clone and express the proteins, and then biochemist skills are needed. This is basically the users. Who are the people who use it? And the biochemists to purify and crystallize the proteins, and then some physicists are interested in, because this, as you know, the biophysicists they were the physicists who were 30 years ago doing physics, then saw the light and came to the biology, including myself as a pure chemist, and I became a biology person, because that's where the future was, right? And then finally, most of us run Linux operating systems. People need to be comfortable running scripts, having command line interaction. That is very needed. And then we need to help the students learn and feel comfortable with it. Even though the Linux operating system has changed over the years, it looks like more a window-like, but still, when they run into problems, we need their help, right? I mean, we can help them, yeah. And as I said, this is once again, I'm just going to repeat here, if I'm just going to forward, you all know this, the biology interaction of, that's why I said it's very, very, very interdisciplinary. If you go to flow cytometry or imaging or genomics, this level of interdisciplinary thing you don't see in the users or in the people, PIs, right? And then, okay. This is another feature. You see that the projects are very long-term, and typically between somebody having a protein ready and crystallization, and then collecting data, multiple failures, and then by the time they solve and publish, it could be 2 years or longer, right? We need to keep the interest, and we need to know where the projects are going, and then we don't want them to be lost, and therefore we want to, at the same time, we want to make sure the charge we do for them is modest, right? And then there's always constant feedback between the students and the PI, and that's where we come in, and then there's always re-engagement and training. Somebody comes there, they start collecting the data, then they come back 6 months later when they have a new protein, which first time it did not crystallize, first time it was not diffract, or the data analysis was back. They go back, retune all of it. Therefore, we have to re-engage them and retrain them, right? That is a kind of a mindset where core facility people, we need to reach out to them, hey, what's going on with your project, right? And therefore, this is where the training part of this comes into picture. For example, what we looked at is there is as a core facility staff, we want to be engaged. At the same time, if the people are not coming to you, somehow you have to reconnect with them. As I come from a university with education as one of the main focuses, therefore what we thought about is about maybe 12 years ago, as the graduate students come, I work in a biophysics department, our students come from wide variety of backgrounds, right? They want to do the structural biology. They join either biology faculty or physics faculty. Even our PIs come from a different background, right? And then, therefore, but the important thing, not only you run as a structural biologist, as a core person, you are focusing on the training. What we have come up with is what is called, one of my colleagues, Peter Randolph, is here. We came up with about 12 years ago what is called boot camp. What we do is, as the students come in, we have about 5 core facilities in my unit, and then we take the students, and then for 1 month, we train them. Basically, what we do is, Monday morning, they may be coming and setting up a crystallization tray with me. The afternoon, they may go to Peter Randolph, and they learn the light scattering, circular dichroism. They may do a project. The next day morning, they go to a protein protein purification or protein expression facility. They learn how to do. Then the afternoon, they may be going to a computer person to learn their coding and what do you call it? Script. Go ahead.
May I interrupt?
Sure, yeah.
So when you do these boot camps, I assume you do them once a year.
Yeah, exactly, yeah.
How do you time them? Because in our case, students come and go, join labs. How do you catch them that they really kind of get the boot camp at the beginning when they need to learn it and then are proficient in carrying it on?
Absolutely. What we have done is, actually the students even do not know who they are going to check their PI. We catch them the moment they arrive on the campus. Typically, the graduate students, in our case, come in the first week of August. The classes typically begin the last week of August. That is the 3 to 4 weeks we capture them. That I was just going to follow up on, I'm going to say. What happens is, as the students come, we kind of make them go through this 5 or 6 different boot camps of mass spectrometry, cryo-EM. We don't have NMR because the NMR is in the magnet lab, National High Field Magnet Lab. Here, Peter Randolph's biophysical chemistry lab, protein expression, and my facility. They go. No. Basically, what I do is we split the 3 weeks, and then they come morning 4 hours with me. I typically, maybe the afternoon 4 hours they are with Peter, and the other next day 4 hours with Mike, who is my computer person, and the next afternoon they may be working with the protein expression. The third day they go to the mass spectrometry. Then they come back to me. Then I follow up on what I started, and then the next, like that, I have about 6 individual 4-hour training for them. And then we design the project such a way that they will know how to do the wet lab, in my case, instrumentation, and finally the data processing. It's a very simple project which will work within 3 to 4 weeks.
That doesn't interfere with other kind of introductory classes that they have to go through?
We do not emphasize any classes. It's all hands on deck.
But like in our case, we have actually the students come in 2 weeks ahead of classes.
Right.
And then they have all kinds of introductory days.
We work around those, like the safety training, the TA training.
Right.
Those are all on the schedule too.
Yeah.
This is the boot camp. It's not necessarily you do the projects and you do the equipment, but you don't know what they're going to do long term. So it's just the fact they know what's available and what it can do, so that if they ever come back later, they know who to talk to and what.
Which then they need additional.
And then they would need the training again. Like I'm always assuming, even if they do the boot camp, when they come back in 6 months, 8 months, I'm going to train them again.
Yeah, right.
But at least they know the equipment is there.
Yes. Yeah.
And they know what they can possibly do with it.
Yeah. Susan.
What is your failure rate?
Failure rates in terms of the people not coming back to me? Or,
Or I guess just in the project in general, because to me it doesn't seem like there's enough time for someone to learn that much to be able to do what I'm thinking. I know what goes on in those areas, and I'm like, wow, that's a lot. For them to come in and make a protein and do the crystallization and then do the diffraction, I mean, all of that stuff.
right. Yeah, you're correct. What we try to design, design is simplest of project. I don't know how many of you are crystallographer. There is the protein lysozyme. If I set it up today, I know the crystal is going 3 days from today, right? I know. Right. The idea is to just get them curious about what my core facility can offer to them even before they begin. And going back to Marcus' question, when they get to the rotation of the PI, they already have, this is exciting to me. I hate this. Like it's like you go through a training before you really accept your thing, right? It's like a speed dating, right? I do not want to ever set foot in the X-ray crystallography lab because I did not like what Soma was talking or what Soma is offering. That's fine. They are not going to come back to me. But like what Peter said is if they liked it, the first iteration, they choose the PI based on because they are going to go through 3 labs. At least one of them could be a structural biology lab, right? Or it could be something else. When they go, the PIs appreciate this because even before they get to the lab, I remember Soma was showing me the crystal tray, how it was setting up. They are appreciated that just 3 weeks of kind of getting their feet wet is sufficient enough to pique their curiosity, and they know what they are going to do. Of course, we bring them back. You put as much, many of you know you have your user manual, you have a YouTube videos, everything. Nobody looks at that, right? They come to you, how do I do this? They all ask you. As a core facility people, we know this. They're going to ask you. But at least they know what instruments you have, what software you have, what kind of answers you can give. Yes, Peter?
I was going to say also a big one is they know us.
Yeah, right. Exactly. Right.
They all know our faces and talk to us.
Yeah. We are, as core facility staff, we play a role in, we are kind of psychologists, right? The PIs do not want to know what students do not know, and the students do not want to tell the PI what they do not know. But they come to us in confidence, hey, look, I really do not know how to do, right? That is where your educational and training part comes, because this person may be excellent in expressing proteins, excellent purification, but they may not know the software skills because they are not trained in that area. That is where you come in, you sit with them, and don't worry, we are not going to let your PI criticize you because you do not know, because you have other skills, right? That is what is needed. That is why the structural biology course is slightly different from regular, purely biology course, because we are training people who are very good at one area, but they need the complementary area where they do not have the training, right? That is where we play a great role. And like Peter said, you broke the ice with the new students, and then they do not hesitate coming and ask you. As you know, many times our core facility staff, we do not know what the people know or do not know. They come and give it to you, hey, look, run this, right? We don't know what the sample is, what the purity of the sample is. If something goes wrong, the PA says, the core facility sucks, right? We are at fault, right? But if you knew the student or if you knew the person ahead of time, they have the confidence. Look, I have 95% pure protein. Is this crystallizable? I'll tell you, no, right? Go and purify. The PI, they can feel comfortable talking to us because we don't determine their destiny. We're helping them, right? Whereas if you go and tell the PI, you don't even know this, that is a big problem, right? I mean, they feel comfortable talking to us. We want them to be successful because we want our course to be successful. We want to produce results which are reproducible and publishable, right? That is where we play a role. And that's why we started this. I think I'm taking too much time, I guess, Malady. And then we are working with the course development where, answering Marcus' question, the PIs will be talking the theory and as a full-fledged course for a semester, and they will come to as a hands-on training for the how to do stuff, right? And then, as I already talked about, we have a lot of open source software. Some of you may be familiar with some of this. There's something called CCP4, UK-based one, and Phoenix, which is like a suite of software you start for the data processing and lots of steps. And it is sometimes opaque and you don't know how it runs. That's where we play a role. That is, and there are many methods to solving the three-dimensional structure using crystallography. We can help them out. And then core facility staff play a great role in helping them, which will work, which will not work. And then, as I said, I already talked to you about that. We play a liaison between the faculty and the students, and then sometimes we take the senior students or postdocs and we link them up and then guide them to have a successful project. Finally, if they are successful, we are successful, and we can provide the help. And this is, I'll quickly go through. Do I have time? Yeah, okay. This is the other part of this, which almost everyone will agree. This is the data was collected in 1994, maybe 1999. You all know this. There were one PhD student have one structure in his thesis. They are done. They graduated and went away, right? And we worked on this 3 years, and then we had 3 dozen data sets, and we solved it to maybe 1.5 angstrom data, right? I mean, this is the time the cryo started coming, like the cryo people. This is the early 1990s. Before that, we used to keep it in a capillary and collect data, and then we collected data. Now, what's happening is, currently the students have multiple data sets, and maybe I even have a slide. Sorry, I don't have the slide on this, but you know, there are multiple structures they have to solve. And then we are moving away, my facility is moving away from doing this locally. We are doing like with Gabby's national facility. We send it to synchrotron where we have the almost 100,000 times more flux of X-rays available. What used to take 1 week, the data you saw in 1998, we collected 1 week, right? 1 week of data collection. Now, the data collection happens That's it. That fast, right? Basically, you put it, and everything is automated. We don't even have to go to the beam line. And 20 years ago, we used to go to the beam line. We don't even have to go. Everything is done by robots. I don't have a picture of it. Basically, you just go in, finishes, and the data is even processed while you're collecting and screening your samples, and they send the processed data back to you, right? It's like you put something comes outside. Once again, this is where we as core facility staff have a great level of training to tell. Just because you put something and something comes out is not really the result. You have to learn how to do this, right? Of course, there are problems and there's also opportunities. We are helping them with data processing, data interpretation, depending upon the PI and the students, whether we involve as people who are just helping them or get involved in the project where you are either co-author or just acknowledgement, or you just even acknowledge the RRIDs for your facility, right? Depending upon how it works, right? The things have changed after the Human Genome Project came. All these automations have come. She will talk about, I think, some of them, but there's also an opportunities, right? And as I said, because my facility is kind of the X-ray part of it is shutting down, I'm expanding to give more like crystallization robots, and we are making crystallization custom buffers, and then we are trying to store the data, and then work with the national facility to coordinate sending the sample and collecting data and processing all those. Those are the things I do. However, maybe in 2 or 3 years, even that will go away, and then I may have to sunset my score. Because some of the challenges are almost impossible to overcome, right? For example, the X-ray beam you see is almost a million times more flux, right? How do you compete with that? You can't build a instrument which is as powerful as the X-ray beam is. The one with the blue line is what I can offer. What is the yellow line? It's a logarithmic scale on that side. I mean, orders of magnitude higher. You can't do it, right? And in terms of the size of the beam, the smallest I could offer is 70 micron. The crystals are very, very tiny. The National Synchronous Facility offers you 5 micron. How do you compete with that? There's no way I can compete. And then similarly, that's why we are actually helping them and going from home source, sometimes to regional centers, sometimes to national labs. And we have some challenges. As I said, I have a skill, and I could either sunset my core, or help in somewhat related diffraction-related facilities, like there are single crystal, small molecule places, or powder diffraction, or move into material science, depending upon the facility, or electron diffraction, because the techniques and the ideas are very similar. Otherwise, basically sunset my score and then go on a retirement. Okay. As I said, some of them we can do, some of them we cannot. This is a summary of what I gave it to you, in the talk. I will wait until Josh and then Gabby talks, and then we have a conversation. Thank you very much. I appreciate it. I think I spoke more than 20 minutes. Why don't we, Josh, go and yeah, that way everybody has a chance to talk. How do we switch this? I think this is the, okay.
Oh, how do I do this without breaking it?
The green arrow is the one to advance. Yeah.
Oh my God,
That's okay.
this isn't the right one.
Oh.
No, I have a laptop. Could you go and I can set it up? Sorry. Oh my
That's okay.
gosh.
That's okay. It's
Things happen.
things happen. Do you have it on your thumb drive?
Yeah, I got my laptop.
Oh, okay.
Sorry, this is real bad.
No, you're all good. All right, slight change in plans. I will be speaking. All right, so I'm going to talk a little bit about training opportunities at the National Crystallization Center, where I am the operations manager. And so what we do at my facility is specifically focused around crystallization. let's see. Oh, this works. Okay, so as Soma kind of hinted at, there are many, many steps in the process of crystallizing a protein and obtaining a structure that can be used for hypothesis generation and determining mechanisms of protein function and things of this nature. But of course, we start with our protein sample, screening many different kinds of chemical conditions in order to find a crystallization condition. Once you have your protein crystal, you can use these synchrotron sources that Soma discussed to collect these diffraction patterns from the crystals, and then after phasing, using some of this open source software, you can actually solve your structure and be happy with your life. But we're really focused on this screening portion because this is a huge bottleneck in protein crystallography. And it's still something that we have really no way to predict how, where, when it will happen. And so this is really what we're focused on at our facility. And I do just want to focus on, or highlight the fact that crystals can be used not only for X-ray crystallography, but also techniques like X-FELS, X-ray free electron laser diffraction, neutron diffraction, and microcrystal electron diffraction, which is a kind of new and exciting technique. So who are our users? We are a national facility, and we're not necessarily associated with any specific research university, but our service is really for anyone. So people that I have had as users, maybe they've already tried all the screens in their lab. Rather than buying another $400 screen from Hampton, they'll send it to us, and we can put it through our 1536 screen. If you manually set up all your trays, you probably are in the early stages of getting carpal tunnel, and so you send us your sample. If you manually inspect all of your wells for crystal growth, our service might be of interest because we have an automated imager, and you get images of your crystals during the growth period. If you don't have advanced robotics or imaging, it would almost be impossible to set up an experiment like we offer. And if you need fast, easy access to screening a wide range of crystallization parameter space, this is the kind of service that is perfect for you. So really anyone can be a user, and we do have users throughout academia, government, nonprofits, and industry as well. So we have been running for over 20 years, and the reason why is that we were established during the Protein Structure Initiative when it was really heavily funded to try and understand the structures of these proteins that we were sequencing from the genome. But recently we actually received the NIGMS National Resource Funding to become the National High Throughput Crystallization Center. And I think this came at a really great time because even though the Protein Structure Initiative ended, I think in 2016, something like that. Now we're seeing more and more of these X-ray cores actually going out of business, and so people are looking for places to be able to screen conditions for growing crystals, and we're here to sort of try and fill that gap. We use a fee-for-service operations model with federal funding support for non-proprietary samples, and I'll sort of share some of this information with you. We've calculated that the cost of setting up our experiment would be around $2,000, but with all the federal funding support, we can offer it to academic users for about $400. So it's a really heavily subsidized experiment. So we do send surveys out to our users requesting feedback, and one of the things that we noticed is that people were really interested in additional training and noted that it's exciting that they don't have to necessarily be an expert in crystallization, but they can still obtain preliminary hits, which are useful for things like grant applications. So increasing the accessibility and training is a positive. Users are also looking for optimization and follow-up with these crystals. So just because they have the ability to produce the sample that they send us for crystallization doesn't necessarily mean that they have the resources to be able to replicate these hits and access beam time and everything of this nature. So we really consider training and accessibility to services and expertise to be a really important component of any research service facility, and we're sort of in a unique position as and part of an independent nonprofit to be able to fill some of these gaps. So I did want to mention one way that we're trying to reach out to our users is through training workshops for crystallization. We had our very first virtual user meeting last year in March of 2023 when we had a little bit of sort of talk about methods and technique, but then also some talks from some of our users about how they were able to leverage the hits that they got from this experiment they did with us into their larger scope of their projects. We're also planning virtual crystallization lectures and on-site workshops this year. May 23rd is our virtual one, and then we have actual on-site workshops in June and August, which we're really excited to actually get people on site and using some of our resources. We do have web-based tutorials and training, which I'll talk about a little bit more in the next few slides. But one thing that I'm super duper proud of that we recently implemented is that we've actually reduced the rate for our high throughput screen for under-resourced institutes and investigators, minority serving institutions and PUIs, down to only $100 for the screen. So we're really trying to lower that barrier to access and make crystallization accessible to labs that maybe have not done it before or are at institutions where they don't have access to a crystallization core. New pipelines in development, we are trying to address that users are interested in optimization, scale-up, and harvesting. And then outreach is another important component. Because we are not affiliated necessarily with any university, we need to kind of be pushy and let people know that we're out there. So we do a lot of social media, presence at meetings, hence me being here, and then one-on-one meetings with users to really understand what their problem is and how we can help them achieve their project goals with us. So a little bit more details about our experiment. This is our high throughput pipeline to find crystallization conditions. We use a 1536 microbatch under oil experiment. And so this really minimizes protein sample volume for our users. They only need to send us 375 microliters of highly pure protein. So for some labs, this is not achievable. Other labs, no problem. But we try to make it as accessible as possible to screen through the chemical space. We provide the robotics, the imaging instruments, and the expertise. So our users often reach out to us like, hey, is my protein concentrated enough? Well, tell me a little bit about it. So we have a back and forth with our users. But importantly, this screening results in a minimum of about 14,000 images per experimental plate. So if you're going through this manually, it is going to take you quite a long time, but we have a way to address that. We monitor the images for this experiment over the course of about 6 weeks. And so we found, at least in our setup, that 6 weeks is about the lifetime of the crystallization plate before it starts to dry up. And so you get a cocktail-only image, so before protein is added. You get day 1, week 2, week 3, week 4, and then week 6 images. And our users then can take this image data to leverage into optimizing these crystals for sending to the synchrotron or whatever experiment they're proposing. So from user feedback at the HTX Center this year, we were really curious if our users have actually been able to find crystal hits. Surprisingly, there's not a really great way to follow up with our users after they've gone through the process. We really should include some sort of like exit survey or something. But a lot of the times people will do the experiment and then not look at the data for like 6 months afterwards. So it's a bit challenging. But through this volunteer survey, we saw that a high proportion of individuals who used our soluble screen were successful in finding hits. A little bit less so with our membrane screen, but this is not surprising because membrane proteins are notoriously difficult targets. And in the same survey, we were really interested in whether our users would be interested in some of these services, and we did notice that crystallization workshops is something that our users are very interested in. So we think this is one of the best ways that we can fill this role and sort of help our users achieve their goals and sort of learn about what we are doing at the Crystallization Center. So with this in mind, we are having this hands-on crystallization workshop at the HTX Center. And when we were thinking about it, we were trying to decide what the best thing to have them do at the center is, because we did consider something like lysozyme, set it up. You can see the crystals even within 24 hours sometimes. But we decided that since so many of our users have already done the high throughput experiment, that they could just bring their own samples and set up optimization trays for their crystals of interest. So that's really what we're interested in doing at our workshop. And it's really a practical workshop in crystallization methods, so how to design a grid screen around a hit condition, a couple different formats of crystallization, crystal imaging. We're equipped with not only bright field, but also sonic imaging. So we want to train these individuals on how the additional information from techniques like SHG and UV-TPEF can actually help you identify crystals you wouldn't be able to see otherwise. And then finally, crystal handling, soaking, and harvesting. The goal is really to expose our participants to robotic liquid handling and imaging that's available for crystallization. And the reason why is that a lot of these instruments are simply not available. Even at core facilities, it's very difficult to get the funding, especially for some of these very, very expensive imagers and liquid handling robots. So another thing I did want to mention, if anyone is looking to do crystallization workshops or workshops of their own in structural biology, Soma did mention there are very few vendors, but they are also very generous, and you can make really close connections with them. So a couple of the supporters that we've had for this workshop are Hampton, which is providing discounted consumables and financial support. SPT Lab Tech is bringing some demo equipment. Formulatrix providing additional rockmaker licenses. And then Mitogen is providing things like pins and other crystallization supplies, and also demoing an instrument. So they can be really good partners to work with. It just takes reaching out and making that connection sometimes. So like Soma mentioned, we do have a lot of fancy equipment at the National Crystallization Center. And so some of these things, Soma mentioned the formulator. So this actually pre-mixes your crystallization cocktails and can plate them into a series of different format plates for your crystallization. We have drop setters like the NT8 from Formulatrix and the SPT Lab Tech Mosquito, which is actually what we're using to set up our 1536 plates now, as well as the Art Robbins Griffin. So we really have a wealth of resources, and we want to figure out the best way that we can sort of get the community, especially our local community, in to use some of these resources. Additionally, we do have 2 Rockimager 1000s with Sonic, which is just like an unprecedented wealth of imagers. We also have Rockimager 54, Mitogen Watershed, obviously pucks and shipping doers. So really we have everything that it takes to get you from protein to crystal at our facility. Another thing that we're really proud of is software that we developed to help analyze your crystallization images. So I mentioned before, a typical experiment for us yields about 14,000 images, which would be, a grad student sitting for hours at their computer and their eyes going blurry looking at all these empty wells. But essentially what this software does, Marco Polo, it's based on the Marco algorithm, which is machine recognition of crystallization outcomes. This is an AI-based tool for crystal scoring. It was developed with a couple of pharmaceutical companies and Google Brain, and what they did was just take a ton of crystallization images and train the model to be able to identify when there's a crystal, when there's precipitate, when the drop is clear, and there's an other category as well that it will sometimes classify things into. So for us, great student Ethan Holloman wrote this software that actually makes this integrated with our 1536 screen so that you can easily go through, classify your experimental image results, and immediately find where you've hit in the plate. And that includes, well number information as well as cocktail information. So this can be as quick as 5 minutes to go through a data set. There is a GUI interface, which makes it a lot more accessible to a lot more people, because even though we do sometimes need to use the command line, it is also a barrier to access for some softwares. There is human viewing and scoring available, so the algorithm is not going to get it right 100% of the time, but there is a way to go in and human score as well, and you can output this to PowerPoint so you can show your friends at lab meeting your awesome crystals. And this is open source, so we're very proud of it. You can also use Marco Polo software on your own screens that are not from HWI. We do have video tutorials available, and the software is available on GitHub, but we're very proud of these. They sort of take you through step-by-step how to operate the software. And about 75% of our users say that they do use Marco Polo to analyze their crystallization results. So we think that there's quite a bit of uptake and that our users are finding this quite useful. So to sort of wrap this up, what we're working on in the HTX Center, and I'm open to any feedback too. If you have ideas, please let me know. Workshops, videos, outreach, creating training and teaching opportunities. So especially at the local level, we're currently writing an EPIC grant with a local university, Canisius College, to have undergrads work on a machine learning algorithm using our 20+ years of historical crystallization data. We think this is kind of cool because not only do you have positive crystallization results in there, you also have negative crystallization results. So we think this is something that could really, with the power of machine learning, be useful for even predicting crystallization conditions, but TBD. We want to help students and practitioners have exposure to technologies for doing structural work and ultimately get people excited about structural biology and protein crystallography. And with that, I would just like to thank my team at the HTX Center, and we'll probably have Josh talk if he's all set up, and then we'll all take questions.
I was going to ask you, where are you located? And I saw Buffalo.
Yes, we're in Buffalo, New York. That is one of our issues, is that we're not sort of a central hub. Buffalo is one of those cities where you do have to take like a connector flight to get to. Exactly, and so it makes it difficult for people to actually get out to us. We are the only one. At least there are crystallization centers that accept samples from other users across the country. We're the only one that has the 1536 microbatch under oil format.
So you mentioned that you need 375 microliters of protein.
Yes.
You didn't say what concentration.
Yeah, so that's part of the discussion that we have with our users. If we're going in blind, we just say 10 megs per mil. But if they have more information, if they can do biophysical assays on their protein, we say the highest concentration where your protein is homogeneous and stable.
And then the other question, okay, are you actually looking for only crystals or some of those precipitate kind of reactions?
Yeah, that's a really good question, because obviously, like, whenever you do a crystallization experiment, it's a point on a phase diagram, right? So it tells you information. We're not really working on anything with that right now, but that's one of the things that machine learning, I think, could be really powerful for.
So they are dismissed if they don't crystallize, they're dismissed.
No, actually, Marcus, follow up. She showed you the formulator, right?
Yeah.
That's what we do is, let's say, all the screens are based on some kind of expectation based on the, let me see, on the knowledge database, right? Because there are multiple, basically the screens were built upon past experience, right? What you asked us about, let's say there's a microcrystal, what you call a precipitate could be a microcrystal.
Mm-hmm.
That's where the formulator comes. Let's say that hit is, for simplicity, it's a HEPAS buffer with a certain pH, for simplicity, sodium chloride, 0.8 molar, right? You know that is not the best condition for that particular protein. Therefore, what we do is we keep this as a center and ask the formulator, in this case it's very simple, you can make a sodium chloride solution. If the buffer has 4 components, as she showed you, the formulator has up to 15 inputs you can do. Ask it to build one increase in one concentration this way, change in pH this way, concentration increase on PEG, polyethylene glycol the other way, whatever component it is. Now you had a precipitate, but it could be scanning both sides of the condition and then look for something better than the precipitate. Then
So you are doing iterative then?
if you could, that's what they could do that. If that is so, if it's so important to you, you could do that. That's where the formulator comes, because it will make a custom buffer for you, as much increments of pH or concentration. You don't have to set it up, and then you redo that. You are now moving from a random hit to a somewhat similar closer area, right?
But the default screen is on a set.
Yep, that's correct.
Or whatever.
We do have sonic imaging, though, which kind of helps you discern what's a true precipitate from a microcrystal, because often microcrystals will be visible using SHG and UV-TPEF, so that's helpful for us as well.
That goes into the results.
Absolutely, yes.
Thank you.
All right, Josh?
Thank you, Gabby.
Thanks. Thanks. I had a little bit of heart attack earlier, but I'm alive.
I think if you could stay,
Oh, sorry.
Microphone.
Sorry. How do
or you could use, does this work? You want to wear this?
No, I'm trying to, how do I go to the next slide?
Are you in the presentation mode? There you go.
Just this. Oh, awesome,
Yeah,
thanks. So we're part of a larger Center for Structural Biology. We're one of 6 cores where we work and help researchers get structures and characterize proteins with different biophysical approaches. So this is kind of how the core exists in a bigger ecosystem at our university. A little bit about our core. We were established in 2019 to enable researchers from UNC Chapel Hill to do cryo-EM. So in some ways, we're kind of a little newish, but to some people using our facility, this is kind of a new technique. We're the only cryo-EM core on campus. We have 1 cryo-TM. We don't do any conventional electron microscopy or negative staining TM, so it's strictly cryo-electron microscopy. We have internal and external customers. Most of them are doing single particle cryo-EM. However, we do get people, especially from the chemistry department, where we do cryo-TM of polymorphic objects like nanoparticles. We also sometimes do tomography, and we're starting to do microelectron diffraction experiments. We operate under a fee-of-service model where we provide researchers with services like specimen preparation, imaging, image analysis, and training. And I'm also invested in method development. We have between 50 and 70 users using our core facility per year. And I'm always trying to apply for grants, maybe not at the same time, but just to get equipment and to get money. That's always, I think, limiting factor for us because we're new and we're small. We have a core committee, advisor committee made up of faculty from different departments, including the biophysics, pharmacy, and cell biology department. We have a faculty advisor, a core director, full-time lab technician, and multiple work-study students who help with the daily operation of the core. So this is just kind of a summary of my talk. It doesn't quite make sense. The other slides sort of put it into perspective, but this is just kind of the thoughts that I wanted to express today. These are things that I've learned and I'm learning in setting up the core. So hopefully, I hope this brings something new to people, but also you could think of it as a tale of caution, because not every core is the same. So maybe this works really well for me, but it may not work for someone else, or maybe you think this isn't going to work, and just it's a tale of caution, like, don't do it, Josh, this is stupid, and that's fine. So I think of the core, the purpose of the core defines the workflow. It's not necessarily the technology or the equipment. It's what is the whole, what is the core's function, and how do we do it? The rest is just getting to that point. Easier said than done. The other thing is I try to be fast and flexible and not rigid and slow. This is in part because as technology changes, methods change, projects change. Being rigid and being slow slows up. It's a bottleneck. It's better to be adaptive. Again, easier said than done. One thing I learned, especially when setting up the microscope schedule, is make the cryogrids the commodity, not the microscope beam time. And I'll talk more about it, but this is important, I think, in scheduling the equipment and thinking about like technologies to invest in. For teaching, this is based mostly on my own personal experience, but also teaching people. Reading a protocol is not the same as learning. So when we teach people, I've a lot of times when I show people use VitrBot, how many times it's like, yeah, I know how to use VitrBot, like that. Yeah. You know, that's different than learning something, but a lot of what we do is very technical. So I think having protocols are very good, but if you're reading a protocol, you're limited to the protocol. It's not the same. We're all victims of bad communication. How do we communicate with people better? There's a few things that we do that I think help, but again, this is something I'm constantly learning. The other thing, I think cryo-TM is a technique, not a routine. So when we do cryo-EM at the core, it's a technique, although some of it, what we do is a routine. I think of it as a technique more. So what's the purpose of the core? It's to get structures. And what that means to me is PI or someone will come to the core, they send me an email and said, hey, Dr. Strauss, here's an Eppendorf tube, I want to get a structure. How it happens doesn't matter to them. So with the core, there's two ways we do this. One, the core does the experiment. The other way, users do the experiment. Most of structures determined are user driving the project, but the core, what we do is we develop workflows, maintain equipment, write SOPs, complete experiments for people, but in the side where a person is getting the structure, we will teach users, provide technical assistance, and for data collection, we're the ones who typically collect the data. However, there are people who operate the microscope completely independent. This is what the core looks like. We have a microscope, a cryo prep room, and another room where we prepare grids with sputter coater and do grid fabrication. When I set up the specimen prep room, this is kind of the equipment that I brought in. This didn't happen overnight, obviously, and I'm not going to go through the workflow, but my thought was I wanted to set up the lab space so that someone brings me an epi tube, they could basically prepare the grids, store the grids long-term, ship them. I'm not offloading anything onto the user. We have everything we need to do the experiment. We also do a lot of grid fabrication where we make our own grids that are specialized. One thing we're trying out is cryocycling the grids, where we recycle the cartridges. This is our microscope setup. We have a 200 kV Talos Arctica and a Gatan K3 direct electron detector. The core staff does the loading of the cartridges and the cassettes, and we also do the clipping. However, we teach people to do both, but we don't have the users do the loading. For people who aren't familiar with the setup, the autoloader has enough slots to image 12 grids. Typically, we do between like 1 and 2 exchanges per day. Sometimes we do less, sometimes we do more. Data is collected on different software programs, networked to the computer. A lot of them are routine, and we transfer the data to a supercomputer with a shared file drive where people can access it, transfer it to their workstation, or process the data over the cluster. A little bit about our capabilities. We mostly do protein structure determination using single particle cryo-EM. We can image a little bit bigger things with tomography like viruses and vesicles, but we tend to be more single particle focused. A few things we're working on are developing microelectron diffraction technique and implementing it. We're also working on going a little bit more towards cell biology, where we image cells and organelles and polymorphic objects using cryo-electron tomography. This is our workflow. I'm not going to go through the steps. We use Serial-EM for data acquisition. This is an open source platform, and we like it a lot. It works well for us and our microscope setup, and we also do a lot of custom workflows and writing our own scripts. We also use programs which will interact with Serial-EM to control the microscope. One of the things that I think is very helpful is being able to process the data in real time. We use CryoSpark Live. There are other programs you could use which basically do the same thing, and we use it to check the quality of the data as it's being collected for screening and data collection sessions, as well as benchmarking and assessing the performance of the instrument. We also use CryoSpark for processing the data. There's a couple examples I'm going to show. I think being able to process the data in real time can be helpful for utilizing the instrument. Quickly, one, this is a G protein coupled receptor. This is an example of an overnight data collection where structure went below 3. And we look at the class averages to assess quality. Is this going to be garbage? Is this going to go to high resolution? A lot of times you learn a lot just from processing the data. Otherwise, you don't really kind of know what you're looking at. Here's an example of a garbage sample. It's a GPCR. It was falling apart, and it's not intuitively obvious from looking at the image statistics other than the 2D class averages. Here's another garbage sample of a GPCR. This is an artifact from having too much detergent or not the right detergent in the sample. Particles look fine, but a lot of times what we do is we find a good grid, we set up a collection. If it looks good, we continue to collect or do these little experiments. It doesn't make sense to really collect on stuff like this. Here's another example of a soluble protein with preferred orientation. So during the initial screening collection, we noticed it was preferred orientation. We ended up tilting to get a structure. It turns out this is actually a contaminant. So here's another example. Someone came in, they just wanted to characterize a mutant in a wild-type protein, so we just quickly collected some data, and we're not trying to get a high-res structure. It's more like we're doing an experiment on the microscope. So I find being able to process the data in real time, it allows us to do this. Change gears a little bit, make cryogrids the commodity, not the microscope. Most of the time is spent on the sample. That's what limits the experiment, not the microscope. For cryo-EM, single particle cryo-EM, near atomic resolution is possible with most microscopes that have a direct electron detector and field emission gun. Most of the time we spend is making that perfect grid, which gets you that perfect structure, and it's easier to export the cryogrids as opposed to the sample. Often some samples are unstable, and it's easier just to make the grids and then store them long-term, image them, or send them somewhere else. I learned this when I was setting up the microscope schedule. So initially I had it where people who were trained would just book the microscope in advance, and I found people were just sort of waiting for the clock to hit midnight, and they would book the microscope, and it ended up working sort of, but I felt it could have been a little bit more efficient, and that people were building their experiments around the microscope time and not what grids they had. So we changed it to a queue system where people use iLab, they fill out a form, and one of the questions we ask is how many grids do you have, where are they? And this helps the course set up the schedule. So we kind of, in doing it this way, we could make more efficient use of the beam time. We're all victims of miscommunication. How do we communicate better? One thing we do is we put a lot of information on the core website that's specific to our core. So if sometimes people ask me a question, I say, look at the core website, it's over there, and it saves me a lot of time and hassle. We also have a Slack channel where we communicate really rapidly with users and postdocs. And people and other users. One of the tools that I like using is the simple poll, where if I wanted to get feedback quickly from people, I would just set this up, and people who are on Slack tend to be more actively using the core than others who kind of use it sporadically. So this is a way I could communicate with the user base quick and efficiently. How do we teach people cryo-EM? This is a good question. We tried something new this year. We implemented Merle's principles of instruction. In practice, what we do is we have different modules for the different aspects of cryo-EM. This is more instrument based, but as an example with the VitraBot, we give them kind of like homework assignments before they set up a training session. We sort of give them we make them do like background reading and understand the process. Then we set up a training module where we work with the person, we show them how to operate the equipment, and we kind of provide oversight. So we give them a chance to play, like do the experiment, but make mistakes in a safe, comfortable environment where we could point out what they should do or shouldn't do. And we use different like teaching tools to sort of reinforce certain concepts and how to best practices. And then once they feel comfortable and the core staff feel comfortable that they've done well, we set up an assessment where we watch them do the experiment, and we use a rubric to sort of grade them, so it's more like consistent from person to person. And we're always thinking of stuff new. Like, for example, this is when we had a student trained end of March, and they kind of they did like something really bad. And then technician and I were thinking, let's change the way we teach people. We had this one person where we graded them, and they were kind of like borderline on one of the items. So maybe we need to teach people like, okay, if you do A, B, and C, and you make these mistakes, we fail you automatically, just to sort of avoid these sort of things. So again, this is kind of an example where we're trying to be flexible. Method development, I'm not really going to, I like to have multiple things going on, but I kind of see cryo-EM as a technique, and there's 3 areas that I think of that are subjected to like method development. One is sample prep, another one is data collection, another is image processing. And when I approach this, I think of how do we improve the technique, how do we think of ways of expanding the core and helping users do experiments. The way, because we're a small core and we're often time and resource limited, I try to reach out to people to take advantage of them and to recruit them and to get them excited about this. So a couple of ways I do this is I'll develop a proof of concept first and just sell it to people and sell it to PIs. Hey, look what we can do. I bet this would work really great for your sample. And then I start asking them for things. This could be like, I need some test samples. Are you willing to give us some test samples to develop this? This could be kind of useful for you to get a structure. A lot of times I'll ask for volunteers. It's not a good name, but sometimes we call it guinea pig. Like one of the postdocs came up with this term, where we would want to set something up, like a software or maybe a method, and we don't have time to do it. So we said, hey, look, I have the software on the computer. I haven't run it yet. Would you test it out for me? And you could have first dibs. Also, I'll ask for collaborators, or this kind of engaging in the PIs and the students can help me build a major and minor user list when I apply for that instrumentation grant. I try to spend between 10 and 20% of my time just on implementing new techniques in the core. Also, research, there's risk. A lot of times it doesn't always work out the way that you want, so I think about this before starting a project. And I also have the core staff lead the project, giving them experience in that and hopefully a paper. This is one example. I'm just going to kind of, I can't go backwards. This is just kind of one example where we spent a lot of time tweaking different ways to collect data on our microscope setup. This helped people collect data more efficiently, but also it gave our lab technician was kind of driving that project, and this was his first research experience. So he got to like get his name on a paper, but also you have that experience, but also add to the core utility. Now I kind of want to wrap up. I'd just like to acknowledge everyone in the core, collaborators at UNC, funding, and some collaborators. Our lab tech, I had a really, really good lab tech. He's actually now a business development manager, probably making a ton more money than I am, but we're very happy. For me, like when I hire someone, they often don't have any experience in cryo-EM, and I try to really like make them as good as they can be, and I try to work very hard with them to develop their career. So it's hard to keep people long term, but I'm happy that they go on and be successful and that they're more successful when they leave.
So since we're talking about training, you mentioned that you have them read some material before they do the course or whatever you call it. Are they actually doing that?
Yes, and is this working? Can you hear me? So I kind of want to just take a step back. I know I'm not trying to dance around your question, but I think there's two words, training and teaching. They mean separate things. in the way my brain works, training is kind of like martial arts or like the movie Star Wars, where Luke Skywalker trains to become a Jedi Knight. Some people, when they do cryo-EM, I want them to be Jedi Knights, like like people who work at our core and some of the students and postdocs. Not everyone wants to do that. So a lot of times when we teach people, it's really just showing them how to like operate equipment or providing. We're not really we're not investing a lot of time. It sounds bad, but like I feel like there's these two routes of like teaching and learning. So training's a little bit than teaching. Teaching I think of as more like broad concepts, understanding more of a process. So some people don't do the reading, and my thought is, well, I'm at a university. My feeling is, as an educator, my job isn't so much to like look over their shoulder at that level. I'm assuming you did your due diligence, you're a good student, you're gonna read the stuff. I find people who don't prepare ahead of time, like you find out really quickly on site. Some people just like, they just ace it, and I think that's fine, and some people absorb and material and concepts differently. So I don't check. I think if I wanted to, I could come up with a way to follow up on that, but I don't feel I need to at this point, and I kind of don't feel that.
And I totally agree, as long as they can do the job that you ask them to do. But if they can't because they haven't prepared, do you fail them?
Yes.
Okay.
We say we need a couple things to work on. We sort of just say like, okay, these are some suggestions. Sometimes it's not necessarily understanding the concept. Sometimes people just can't pick up grids, and it's not their fault, and we try to work with them. Like, so yeah, but I failed people.
Okay.
I have a question. For the crystallographers, how has cryo-EM impacted your crystallography core?
You want to go?
No, you go first.
It is hard. What's happening is most of the original crystallographers are slowly moving toward EM, and then it is hard for us to stop because there are some projects where they need our help, right? There are some projects they need our help. There are certain things where they don't need us because they are big systems, and then you can show multiple way the molecule is there, right? It is hard for us, and I know the technology is going to be going further and further, and the whatever 2 angstrom is going to be one day 1.5 or whatever is going to happen. We know that this is going to happen. At the same time, there is a place where for example, AlphaFold, right? You all know about what AlphaFold is, right? It is AI-generated DeepMind thing which predicts the structure of the protein. If you look at the protein data bank, there are 1,800,000 AlphaFold predicted structures. That's humanly solved maybe 250,000, right? But we know there is a limit on what it is. I mean, it's the unique things what we do. There are still, however, people who want to do the crystallization project because they are small. Right? That's where small in the sense slightly bigger than NMR, slightly smaller than EM. They will still fill into the niche area. I hope I don't know the answer to your question. That's kind of thing, right?
Trust and verify.
Yeah, trust and verify. Yeah, trust and verify. Yeah.
Yeah, I think there's always going to be a place for X-ray crystallography in structural science. I don't know if everyone will agree with me, but I think there is. And I think that there's a lot of competition now for time on the microscope, and it's a whole different workflow that you need to employ. And so, you should be thinking very carefully in your lab about which route you want to go. Do you want to do a combination? We have a lot of users that come to us with structures they want of proteins with ligands, for example. Right now, X-ray crystallography is probably the best way to do that, to get high enough resolution to actually see the contacts that your ligand is making, with the exception of really large proteins, obviously. And another way that we've sort of leveraged the growing popularity of cryo-EM at our center is that we have obviously really strong crystallization experience and really strong cryo-EM. So we're able to tackle things like micro-ED, I think, with expertise on both the crystal end and the microscope end. So it makes it more tractable for us to be able to do projects like this. And I think that's kind of like the dream team for micro-ED, is to have both crystallographers and microscopists in the room. So that would be my answer. I think, it's always going to be a technique that people will come back to, but that's my hope.
And following upon, for example, unless people send the samples to national centers, NYSBC and things like that, the Western Pacific Northwest Center, it is still prohibitively expensive, right? Because, as you know, we have a Titan Cryos, the service contract is $300,000. That is pretty expensive, right? Whereas, of course, that is where we are moving, right? That's what X-ray crystallography moved. We are no longer maintaining home sources. We have moved into nationally funded beam lines. APS, Argonne National Lab, has upgraded 32 beam lines with the billions of dollars of taxpayer fund. It's opening up, right? When you keep pushing things, people find something to do with it. That's where the innovation and the kind of, like you said, when you're forced to do this, we will find a niche. And I don't think, people don't take it wrong. There was a big push about this because of how it was in 1990s X-ray crystallography. EM is the thing to do now, right? Probably it'll turn out to be as good as X-ray crystallography, but until then, we still have some business to do.
Proteins under 50 kilodaltons.
Yeah, exactly. Correct. Yeah.
Yeah, that's like, so you're talking, I don't do X-ray crystallography, I only do cryo-EM, but still, like that whole workflow of, attaching to scaffolds and everything, like, if you have the expense and the time, whereas now crystallography, if you're sending it to like national beam line, like the cost there is much.
Practically zero. We call FedEx data collect. And just to follow up on what she was showing, we can actually take the plate now and put it on the beam line, right? You don't even have to scoop the crystals. You can put it on the beam line. Also, there's a serial crystallography where you can do basically a stream of crystals going through the beam. And we were originally single crystallographers, right? Now it is very similar to low crystallography, where you see all the orientations of the crystals going through the beam line, because the data processing and the computer software has developed so much. What used to be a problem for us is actually coming to help us. These are the things, as I said, as you're pushed to the wall, you figure out a way to solve the problem, right? This is some of the things we are doing, even though it may not be happening in my home facility, but it's happening in crystallography, right? Femtosecond crystallography, things like that, right? Is in SSRL in Stanford, yeah.
Question for Josh. So, operational wise, do you train every user?
No, some of the people who use the core just send us samples and we do the experiment for them. I say users like they're using the core facility, so it's not everyone. In terms of breakdown, I would say about like, I guess there's different ways to quantify it. One could be the number of people that send us samples that we do the experiment for. The other way of quantifying it is how much beam time you use. So I kind of have these difficult conversations with some of the people in the pharmacy who, I think they use the core a lot, but like, no, we only use it this many hours. I'm like, look, like you're using it, and we get this stuff done. To me, that's good, and you're using the core. So I think it's kind of a little bit of a mix in terms of the breakdown. I'd say it will range. I think there's probably like 50% of the people are getting trained at some level, whether it's using the VitraBot, using the microscope, or doing some sort of image processing. Most people I find get trained on like the sample prep and then the microscope at different levels of operation. And most people who are doing cryo-EM in a cryo-EM lab will learn how to do the processing either in their own lab or through interacting with the core at some point.
Josh, I think I just want to follow up on, for example, how do you get acknowledged, say a person, like you said, sends you a sample to solve the structure, they just pay and they don't acknowledge you, are you part of the project? How do you determine? Because that is one of the biggest problems we face as core facility manager. Where do you draw the line? Do you talk to them early or you get involved? Could you elaborate on that?
My approach is structural determination is an intellectual achievement, not a technical achievement. My old boss would say that a lot, Mario Bornia. I take that, I kind of use that at our facility. I don't feel like my job is to publish a lot. It's just to sort of run the core. What I typically do is if I tell people, like, these are the routine things that we consider routine. We're not authors on the paper, just mention the core, that's a plus. If we're collaborating, we're doing non-routine things, and we have a meeting and we discuss roles of each person in the project. Typically, I try to get the lab techs or the students on the paper, or I say, hey, look, they're really good at doing this. If they'll do the experiment for you, I want their name on the paper, and we have a discussion afterwards. Sometimes it gets a little dicey, but that's the nature of collaborating, I think. But I try to follow the NIH guidelines.
All right, well, if there's nothing else, thank you all for coming, and introduce yourselves to each other, because this is our little structural biology community and we got to stick together.
Yeah.
Enjoy the rest of your conference.
And then put in comments for 2025 if you want to see more like this. Please, there's a board in the main area, what you want to see in 2025. I know, yeah. Right. That is the perception we want to change, because we want to improve this as, I speak both ways, right? Because I'm a crystallographer and also ABRF board member. I want this to be expanded. Thank you very much for